afternoon, everybody. So welcome to today's physics colloquium. So it's my pleasure to introduce Professor Ibrahim Cissé from MIT, Department of Physics. For those of you that saw his condensed matter seminar a few months back, I hope you'll pardon my repetition of a few interesting biographical details. So Ibrahim has an extremely interesting life history, and if you have a chance, I couldn't recommend highly enough to talk to him about it. He grew up in Niger, where he basically went to French schools, and at the age of 17 decided to buck tradition. And instead of doing the usual thing, which was to go to Paris for his continued studies, he decided to come to the US, where he went to North Carolina Central University. And you might find it interesting to go look up that college. And while he was there, he decided to do a summer internship at Princeton, where he worked with Paul Chaikin. And he did a very famous experiment in which they basically looked at the packing of M&Ms. And it's a paper that's in Science magazine that's super highly cited. And it's kind of funny because Chaikin says of it, that many undergrads had stumbled upon this thing and not really figured it out. And then Ibrahim came along and figured out how to basically understand the nature of the context of the M&Ms. And I think that that's a great story. So after that, he went to Urbana, where he did a PhD in physics. And that was followed by a postdoc in Paris, true to familial form. And then he went to the HHMI Janelia Farms, and then to MIT as a physics professor. So what he works on is sort of the interface between interesting questions and how cells make decisions and the kinds of methods that we're used to in physics for trying to actually make those measurements. So without any further ado, it's really a great pleasure to welcome you back.
Thank you.
So nice to see you.
Thank you, Rob. It is indeed exciting to be here to tell you about the efforts in my lab at MIT to bring in this very high-resolution microscopy directly inside living cells and look at how biomolecules actually do their function. And in this case, it would be in how our genome, genetic information, is decoded from DNA into RNA. And we'll talk more about that. But first, I really wanted to acknowledge the people, as they say, who do the work. I feel like as a faculty, I'm really the very lucky custodian of the group of extremely talented students and postdocs that you can see here. So this is the current group. And today I will try to highlight the works of some recent alumni. So Wong Ki-cho, Jan-Hendrik Spiele, and if time permits, Arjun Narayanan's work as well. The general interest in my lab is to understand how cooperativity emerges in systems that have very weak and transient interactions. Problems that involve many different components, as you know, can be some of the most intricate to understand from first principles in physics. And this is especially true if the different components have an interaction between them. So typically we tell students, right, if it's a two-body problem, we may expect them to solve it exactly. If it's an Avogadro number of components, they can use statistical mechanics to make inferences from that. But biology operates in the in-between, where our physical intuitions tend to fail. However, even if these interactions are of very low affinity, sometimes a system can exhibit a collective behavior where the whole can do more than the sum of the individual components. This kind of emergent phenomena, we know control complex dynamical systems at the microscopic level. And when we started our lab about 6 years ago, our bet was that that was likely how the genome also self-regulates, if only we had the capability to go down to that level and uncover these processes with sufficient quantitative details. So what I tell students usually is that there are collective behavior in living systems that you can observe even by the naked eye. So for example, an ant colony carrying a large amount of food, or a school of fish that dynamically reorganizes into cluster to avoid being food to a predator here, coming on your right. But the ones we'll be talking about today in terms of scale require us to start from the human scale, zoom in first 1,000,000 folds, and that brings us inside the cell to an area around the cell nucleus where our genome is packaged, where many of these processes that I will talk about take place. Some of you with the best eyesight might be able to make out an oval shape in the middle here. Are you? Okay, so that's the cell nucleus. But this picture becomes very blurry very quickly. That's because you're trying to do extremely rapid imaging at the very limit of optical microscopy. So the techniques my lab would adopt, and that we'll talk about, and further develop for our specific purposes, would be so-called single molecule-based super-resolution techniques. We'll talk about that. They go by acronyms like STORM and PALM. They break the optical diffraction barrier to reveal in finer detail the organization of the biomolecules within. And in particular, what we will care about would be some of these clusters that are formed. We can take some of the largest clusters, zoom in another order of magnitude, and notice how now the scale bar is one wavelength of visible light. This is 500 nanometer. At this scale, you can see not just individual biomolecules, but when the molecules come together to form these clusters. In fact, when I first showed these pictures to some of my colleagues in Astro, they told me, oh, this looks like the stars and galaxies that they image also. But of course, this is not the very, very large, but the very, very small. We'll also talk about the molecule of interest here. It's a molecule that's called RNA polymerase 2. It's a mouthful, so we'll just call it POL2 for short. So what does POL2 do? Well, POL2, in my opinion, is one of the most interesting proteins in all of biology, in all of our cells. It's the molecule that decodes the genetic information from DNA into RNA. Many of us may remember from high school biology that the genetic information we inherit from our parents in the form of DNA, which is this series of letter codes ATGCs in this double helix. It must first be copied into an RNA copy before it's made into a protein. Now, if you're like me, before I started in this field, protein is just some kind of food supplement that people take if they want more muscle. But proteins are really the biomolecules that do all the energetic work inside the cell, so they can consume ATP to do more work. They do much of the heavy lifting inside the cell. Well, then the protein that then goes and binds to DNA to decode the DNA into messenger RNA is the protein that's RNA polymerase 2 in all eukaryotes, including us humans. So if you ask how POL2 works inside the cell, well, there is a lot that is known. In fact, you can go on Google and try to find cartoons like this that depicts at least how we teach about how this process works. So represented here, for example, in pink is the DNA in double helix, right? And the polymerase will need to be so-called recruited, and we'll talk about what that means. So the polymerase may not know where to bind, so it's assisted by a general factor that first bind. Once this is bound, then you'll see the polymerase that will come in green here, right? Alongside dozens of other cofactors. Of course, they're diffusing, but in this cartoon, they will just magically fly in from the right. Once this machine has formed, so-called the pre-initiation complex, then a distal region is believed to loop around to come in and know exactly where the polymerase is. Touch it, release it, and the polymerase will then track on the DNA like a train on a railroad, unwinding this double helix to reveal the series of letter codes ATGCs, which it copies into an RNA copy coming out in yellow there. Now, however elaborate this cartoon may seem, it's still just that. It's just a cartoon, right? It's too simplistic and perhaps even wrong. The reality is that there is no method of microscopy right now that would allow you to look at processes, dynamic processes like this with sufficient detail. And the reality has to do with what we say in our field when we claim that we can see single biomolecules. So for that, we have to go back and look through some of the technical limitations for the techniques that we will use. These techniques, of course, come from physics, so we have to think about what the physical mechanisms that give rise to these techniques are so that we can think a bit more carefully about what those limitations are. And everything in this talk is about now trying to overcome some of these limitations to try to see what we may not have seen before. So the best approach really, most biologists will tell you to look at the dynamic processes inside living cell will be to use a fluorescence microscope. So the term fluorescence itself is really a term that was coined by physicist George Stokes. And it's a quantum mechanical phenomenon illustrated in this Jablonski diagram. It's properties of certain materials that are able to absorb blue photon, go through an excited state, relax through a lower energy level, and when they finally relax back to the ground state, they will emit a redshifted photon. So if you absorb blue, you may emit green. But in fact, Stokes himself did not first call this fluorescence in the main manuscript, right? So he describes this, that he's studying the remarkable phenomenon that was first discovered by Sir John Herschel in a solution of sulfate of quinine, that the solution of quinine, though it appears to be perfectly transparent like water, when viewed under certain transmitted light, would exhibit certain aspect of a beautiful celestial blue color. Now, when Stokes wrote this, he called the process dispersive reflection. And then this is a note to the students who like to read papers without reading, the footnote. So Stokes had a footnote in this paper about this dispersive reflection term that he coined. He said, I confess I do not like this term. I am almost inclined to coin a word and call the appearance fluorescence from the fluorospar minerals analogous to the opalescence, or rather, call this from fluorospar, but analogous to opalescence, which was called from the minerals, right? So I would say, right, we're lucky that this dispersive reflection term did not sustain, and now we're just calling it fluorescence microscopy. Now, if this can happen in minerals, you can bet through evolution that, there might have been some living organisms that have evolved to use the same physics to absorb and emit light. And in fact, that's what marine biologists who were asking the question, why do this jellyfish, the crystal jellyfish, glow in the dark? And what they were able to ultimately discover, and which really changed the course of fluorescence microscopy, and especially allowing to do fluorescence microscopy inside living cell, is their discovery that there is in fact a gene, right, that is a sequence of DNA, that is made into RNA, that is then made into a protein, and now the property of that protein, which is a so-called green fluorescent protein, is to absorb blue light and emit green light. Now, the molecule itself has a Stokes radius of 1 to 2 nanometer. But of course, when we say we're looking at that molecule, what we really mean is that we're looking at the light emitted from that molecule. Light is a wave, so it will diffract. So in reality, what you see is this big blob that is 2 orders of magnitude larger than the actual size of the molecule, right? So if you want to make out multiple molecules distinctly, they better be separated by more than the wavelength of light, or more than 100 times their actual size. Now, I know that there are students in the audience, right, and that we are in the Feynman Hall after all, and any basic process that you would want to understand in physics, probably Feynman has found a great way of explaining it. So there is a whole chapter on diffraction in the Feynman lecture, and it's really this property that even if you have a finite length emitter, then if you look at the projection from that finite length emitter, you're going to get this kind of so-called airy disk pattern where you get huge constructive interference in the middle and the first nodes of destructive interference right here. Of course, the uncertainty or the broadness of this constructive interference central lobe is determined by where this first destructive interference happens, and that is of the order of the wavelength that you use. So your uncertainty is still about the wavelength of the light that you use. And this was immortalized in optical imaging by Ernst Abbe, that in fact the resolution distance that you get is the wavelength divided by 2 times this n is the index of refraction of the best lens that you can use, and then this angle sine theta is the aperture. So about 1/2 the wavelength of the light that you are looking at is about the resolution. And this is true of the largest telescopes that you can build down to the smallest microscopes, and even the optical devices that you have in your cell phone that you take pictures with. So then you will say, if we want to resolve things in biology in the nanometer scale, then why don't we just use an electromagnetic wave of a lower wavelength so we'll get a higher resolution? But remember, if you want to do this inside a living object, what happens if this is sunny California? What happens if you must go surf for a long time in the sun? You need some protection, right? And that's because already if you come to the UV range, about 300, 400 nanometer, you're already starting to do cross-linking that will kill your cells. So we use visible light because it's non-invasive to the biological processes. So then you may think, okay, if we're using visible light, then we're limited to this diffraction limit. But then people in our field a few years earlier had the simple idea that, well, the opposite is true, right? If you can separate your localizations, even in time, you certainly can localize the centers with nanometer accuracy. And that's the principle behind single molecule-based so-called super resolution techniques. They use the fact that you can make these molecules blink one at a time and localize their centers with nanometer accuracy and resolve pictures at the bottom that are much sharper than what conventional pictures can resolve at the top. So these super resolution techniques can go by many acronyms. You may have heard of PALM, which stands for Photo Activation Localization Microscopy, or STORM, Stochastic Optical Reconstruction Microscopy. Those acronyms will not matter. For this talk, we can assume they work under the same general principle. And I would say that the people who came up with this idea fairly recently were handsomely rewarded by the Nobel Prize, but also by the Breakthrough Prize. So how does it actually work more practically? So let's try an experiment here. Would you guys play along with me? Yeah? Okay. So here's what we're going to try to do. Imagine we're all biomolecules, and I'm trying to resolve the fact that you are all neatly seated in this kind of rows. But all I can do is not use my eyes, which can clearly resolve you, but I'm going to turn around and use my ears. So we need something that the whole group is going to shout out at once. So we need to pick a word. Caltech. Okay, great. So here's what's going to happen. I'm going to turn around, and on a count of 3, everybody is going to shout Caltech. Are we ready? Okay. So, and from that shout, I'm going to have to try to resolve how you're organized, okay? So let's try this. 1,
Close your eyes.
2, 3. Caltech. Okay. Would I be able to resolve you? No. Okay. So here's something else that we can try. I'm going to turn around, and then on the count of 3, if your birthday is within a week of today, then you should scream Caltech. If not, then you should stay quiet. Okay, so 1, 2, 3.
Caltech.
Okay, great. So we can already tell that there was maybe someone somewhere around here, right? A couple of people around here, and maybe someone somewhere around here, right? By the way, happy birthday. Right? So then we can do that by moving the weeks. There will be some weeks that we will get no detection, and there will be some weeks that we get many detection, and you know, if there is enough stochasticity, then you can separate the localization and you'll be able to reconstruct like that. Does that make sense? So that's actually how super resolution works. So imagine your biomolecules are all organized in some kind of organization, and you label them with this green fluorescent protein. But with conventional microscopy, they all scream at the same time. But then with super resolution, you go to a case where virtually all of them are off. And then you try to make it so that only a few random ones will turn on at the same time. You can then localize those ones, record their localization, wait for them to go off, and then wait for another random set to come on. at no point do you get the full picture in any single frame. But if you wait long enough, right, and this really takes a long time, you may get enough localization. Takes a really long time to try to reconstruct the organization. So, of course, if your biomolecules start to move around, you could end up in the wrong place, right? So, understandably, people in our field say that, okay, these super resolution techniques work great, but really we came to accept that they work only when things are very static, if you must look at them inside a living cell, or you have to fix the cell, meaning you chemically cross-link everything. The cell will be dead, but there is no dynamics, but you can get a clear snapshot of how things are organized, right? But I just started this talk by telling you that things that I'm interested in tends to be collective behaviors that happen below the optical diffraction limit, so we need this kind of techniques, but they also tend to involve a very low affinity interaction, so they tend to be highly dynamic. So our first order of business was to try to find a way to relieve this precondition, do super resolution directly inside the living cell for highly dynamic processes as well. So I will tell you about one approach, come to be known as so-called TC-PALM. It's a time-correlated super resolution approach. And the idea is basically simple. It's to say that if I care about not just where I localize the molecule, but when I localize the molecule, I can use that temporal information to infer on dynamics. So more practically, how would TC-PALM work? So imagine you do the super resolution inside a living cell, and in the nucleus, you see that there is a cluster of pol II here. So if you now look at this cluster of pol II, you can ask, was it dynamic or was it static? So in fact, how would I know whether a cluster is static? I could just do, as a control, the same thing that people do. Chemically freeze everything, and then do the same imaging, and look at the temporal history of detection. The premise for TC-PALM is this. If this cluster was always there, and you stochastically activated and detected it one molecule at a time, if you look at your temporal history of detection, it should obey a statistic that is consistent with the stochastic process. So what I'm going to show you is what the data looks like for a cluster from a chemically frozen cell. So there is no dynamics. So we can see what things look like in one extreme. So this is for a cluster, but in a so-called chemically fixed. So you're looking at here at the top how many detections you get per frame. So it's typically 1 or 0, but sometimes, especially in the beginning, you may get 2 or 3 that you detect at the same time in that region. Is that okay? Then, over time, as you start to get more and more detection, you get a gradual decrease in the frequency of the detections that you get. If you look at here at the bottom, it's the same information that is represented in a cumulant form. So it's a function that will always go up. Every time you get a detection, you go up by 1. But now the slopes represent the density of the detection that you're getting. You get a high frequency of detection from the beginning of acquisition because there was a cluster present from the beginning of acquisition, and then you stochastically activated and detected the molecules, and you go through this kind of gradual plateau. This is the signature for a stable cluster. So now we can go to our clusters inside the living cell and look at, let's say, this pol II cluster, and this is what the data looks like. Notice now in this region, for the first minute and a half, there is virtually no detection. No detection does not mean that there was no protein there, right? It just means that likely the local concentration was so low that the probability of photoactivating and detecting a molecule was negligible, right? So if I take, virtually everybody out of this room except just a few random people, if I try to do that and say, okay, if your birthday is within a week, more than likely I won't get a detection. It doesn't mean there is no one there. It just means that the concentration was very low. But when a cluster does occur, here, it results in a high frequency of detection, And when a cluster disassembles, it results back again to a low frequency of detection. So with this kind of TC-PALM readout, there is actually a lot of information you can extract. The simplest thing is to say you know when a cluster assembled, you know when it seems to disassemble, so you can estimate the cluster lifetime. So I would say this is where it seemed as if we actually shot ourselves in the foot. Because I think many biologists were actually quite happy that we were able to show that clusters existed inside a living cell. Then they were happy that we pushed to try to extract dynamic information. But the problem then is, what is the average cluster lifetime that we see? It's about 8 seconds. Now, 8 seconds can be a really long time or a really short time, depending on who you're talking to. Right? But remember this process where the polymerase binds DNA and then presumably tracks on the DNA. So you can think about the speed at which this elongation is happening. And it's about 2,500 base pairs per minute. And a typical gene size in mammals is about 10,000 base pairs. So it should take several minutes to even finish one messenger RNA. So when we first published this, and this was at the end of my postdoc, a lot of people, I would say rightfully so, criticized the work and said, okay, this is maybe great because it's a clever way of extracting dynamic information from super resolution data. But too bad that what they discovered is something that can have no implication in biology because it's too dynamic, much shorter than what the biological process actually is. You say it's something that physicists would be excited about, right? They care about these highly dynamic processes, but certainly no function in biology. Well, when I started my lab, needless to say, right, I had a bunch of physics students who had a very different opinion, that maybe this could be telling us something that we may not have thought about before in the biological process. So the first thing that they tried to do, they say, well, maybe it's actually because of the activity itself. Maybe it's because it's so functional, it's so active, that it's also so transient. So what happens if you block the activity? You can use a drug that basically blocks the polymerase in the track and prevents this elongation. And when you block that, this is still in living cell, but with this drug that blocks the process, now look at what the cluster looks like. What does it look like? You're all TC-PALM experts now, right? What does it look like? A static cluster. So, in fact, this cluster that would normally disassemble within just a few seconds now become significantly more stable if you just block the process. So we say, there might be something to this, right? That maybe it is doing something, it is teaching us something that we didn't know before about this transcription process. So then one of my trainees, Onkicho, decided to push the microscopy to now do dual color super resolution microscopy inside a living cell. And the idea is this. In one color, we can see the POL2 clusters. What about if we label one of the genes where we know RNA should be made out of? Would we be able to see cluster happening at that gene locus? That's what he aimed for. So without going into too much detail, one of the genes for the experts in the audience that we label is beta-actin, and the approach for labeling the RNA, so this is the yellow piece that is coming out, is this so-called MS2-based approach. The idea is that you will see a lot of RNAs, even single RNAs that are made and diffusing around, but that's not where we care about. What we care about is the source of the fountain where multiple RNAs are being made in a burst. And can you see this locus here? That is just slightly brighter because there is a few RNAs that are still bound and being made. And so we can go in this 2D microscope and focus exactly at that gene locus, and then do super resolution in one color to better localize that gene locus with nanometer accuracy that we focused on. Now, inside that same plane, we have polymerase that is labeled in a different color. By the way, this is what people would normally see with conventional microscopy, but by now you know that we can do TC-PALM to reveal all the POL2 clusters that are in that same plane. Presumably, in that plane, there can be many other genes that we've not labeled, right? But we know at least one of the genes that we've labeled, and we know that locus. So we can ask whether a cluster is happening exactly at that gene locus. And in fact, it is. Okay. So we can see that clusters are happening at least in part on this active gene loci. But now, we know the brightness of the gene locus, and we know the brightness of a single RNA, right? So we can estimate how many RNAs are being made at this gene locus, right? But we can also do TC-Palm to measure the dynamics of the clusters. And we can ask whether there is any correlation between the presence of this cluster, the cluster dynamics, and the RNA coming out of the gene. And when we plot now the cluster lifetime for just the cluster that colocalizes on the gene, and then plot that as a function of the number of messenger RNA that is made, we get this linear correlation, suggesting that although the cluster still lasts just a few seconds, the longer the cluster is there, proportionately more messenger RNA is being made. In fact, some of the experts in the audience would know that this gene, beta-actin, is one of the most well-characterized genes. It's a gene that people know works in this burst. So the characterization had been that for this gene, it turns on once every 5 to 10 minutes. When the gene turns on, it makes, on average, about 4 messenger RNA, and it turns off, and it turns on again. And make 4 messenger RNA, then turns off, then turns on again every 5 minutes. Then you can stimulate the cell, right, with some cocktail that now turns up the gene expression. People knew that this is the response of the cell. Yeah, let me show you this. Okay. Right. People knew that this would be the response of the cell. So this is plotting how many mRNA, what's the burst size? It's about, on average, 4 messenger RNA. But if you stimulate it with that cocktail at time 0, you go from the basal level of about 4 messenger RNA, you increase about 4-fold the number of RNA that comes out per burst, and then it relaxes back to the basal output. This one was known. But what was not known was that there will be a cluster that is forming on top of the gene. So we can ask what is happening to the cluster dynamics, and this is what we see. You're plotting here the cluster lifetime. It's about 8 seconds cluster lifetime. You can see that after stimulation, the cluster lifetime goes up about 4-fold and it decreases back to the basal level. So this was known before. What was not known was this input. If I think of this as an input and this as an output, there are a few qualitative differences, right? For example, the input signal is narrower than the output signal. And this could be indicative of some noise in your system. But also, the peak of the pole 2, or the input, happens consistently about 2.5 minutes before the peak of the output. And this is exactly consistent with how long it will take for a process that happens at the beginning, right? Loads of few polymerases, and those polymerases are traveling at a rate of about 2,500 base pairs per minute before they arrive to the end of the gene where you put this tag and can see the messenger RNA. So all of these start now to make sense. So what do we think is going on? We suspect that the textbook model, right, that the single molecule will be diffusing around and randomly find a transcription site, and then maybe assisted by other factors, then you will start transcribing an RNA. It could still be happening, but it's probably too inefficient, especially for genes that we know the cell is able to receive an external signal and rapidly turn on these genes. We think then for many key genes, they can utilize this transient clustering of the polymerase just when it needs to, meaning when the polymerase is being so-called recruited. So it would increase the local concentration around the gene, facilitate the loading of the polymerase. What our data suggests is that for every 2 seconds that the cluster is there, 1 polymerase is properly loaded. But the cluster lasts just about 8 seconds. So that's enough time to load about 4 polymerases, which will give you 4 messenger RNA. And that's the basal output for the gene. So now you stimulate the cell in a way that then you want to get more output. It's in the way the cell responds is by taking the same clustering that happens, by the way, once every 5 to 10 minutes, hold the cluster n times longer, giving the opportunity to load n times more polymerases, which will then give you n times more messenger RNA. So this simple picture explains everything that we've seen in this study. And explains, for example, why the cluster will happen at the gene locus, and then how you would get this kind of linear correlation between the few second cluster lifetime and the number of messenger RNA. Are we okay? Yeah? Okay. Yeah.
So, a few slides back, you show that when the cluster forms, you can localize in time where the transition happens. You're just looking at one diffraction limited area in that whole field, right?
That's right. So when we do the imaging, we do get the full, in fact, we want this. We want to be able to visualize the full cell nucleus. But when we're doing this analysis now, we have another knowledge, which is where this particular gene is that we've labeled. And so all this subsequent analysis is just for the clusters that are happening and colocalizing exactly at that gene locus.
Does this, so then follow-up question, does this work generally, or does it require some sort of sparsity within those
right.
diffraction limited volumes?
So that's right. So if we try to now say we're labeling, there is 23,000 genes in the human genome, right? And it's estimated that maybe about 5,000 to 6,000 of those are active at any given time. And here we're looking at a single plane, so you're talking about an axis of maybe about 10 or 20 microns here. I know they're 10 micron in depth, but our focal depth is about 600 nanometer. So here we're just seeing a slice. And even in that slice, you can really see that we're resolving a lot of individual clusters. So presumably, under each one of these clusters, there may be genes that are active. But what we don't know is whether there is more than one gene under this cluster. And this is, in fact, something that we are looking at right now. So maybe now pushing the 3-color imaging, where 2 colors are for the genes that people tend to colocalize, we might be able to resolve whether their colocalization results in a co-regulation. And this is one of very active area of research in so-called developmental biology. Yes, a developmental biologist. Yeah.
It was wonderful talking with you. You were here before. Thank you for coming back. Do you know, I don't remember, but you know what controls the size of the cluster?
Yeah.
Because actin is one of those genes that actually is pretty strongly expressed.
Right.
Is the issue at the level of how long the cluster lasts or how big the cluster is?
So that's an excellent question. So in fact, when we did this stimulation, we knew that the output was going to change. And we were hoping that there will be some correlation with the dynamics, right? In the end, the correlation ended up being with the cluster lifetime. And for this gene, that was a bit telling. Because now if you look at how, there are a few measures that we can have with TCPOP. We can look at how frequently is this cluster happening on this gene locus. And that seems to be once every 5 minutes. That is consistent with the initiation rate of the gene. And that did not change. We can also ask how many polymerases on average come into the cluster for this particular gene. Did that change? That also did not change. It was very flat. We got on average about 80 polymerases on the gene. What really seemed to change for this gene was the lifetime, how long the cluster was there. In fact, then you can go do, take some of Michael Elowitz works, right, and try to do, predictions on if this gene actually wanted to reach more maximal output, what would you be able to tune? It turns out actually the cell should have tuned the frequency, because then, the fact that, okay, I hope I'm not losing people here, because this is also interesting. So you stimulate the cell to get more output, but look at how many, there is a maximum in how many mRNA you can get per burst, right? That basically is a physical limit in the sense that you are loading these molecules, and these molecules are going to have a certain footprint. So the maximum number of molecules that you can load depends on the size of your gene. So if you just let these ones go, and then rather than waiting another 5 minutes to turn it on, turn on another cluster and load another, then this thing can look more continuous and in fact get more output rather. It just so happened that at least for this stimulus and this particular gene, what seemed to change is the lifetime. Okay, so moving forward, we thought we'd been too narrowly focused at what happens with the polymerase that gets recruited at the gene and in a so-called region called the promoter. As I told you, there is a distal region, a so-called enhancer, that also recruits its own set of factors, and chief among them is the so-called mediator complex that is believed to loop around, come in and directly touch the polymerase, and that's after the touching that the polymerase can then go. Well, the best studies for mediator recently in the field really had come from a technique that is called ChIP-seq. Okay, what is it? Remember, you can chemically freeze the cell, right? The cell is dead. Then you can chop up all the DNA, right? So then let's say you have something that has a high affinity for the molecule that you want, mediator. So when you pull down the mediator, you will pull it down in complex with everything that the mediator was associated with, including the DNA that the mediator binds to. So then if you do sequencing after that pulling down, then you only see, or you preferentially see where the mediator was bound, right? So that's how people found that for many key genes, it's not just a single binding site that there is, but tandem repeats of binding sites that they call super enhancers. Now, genomic sequencing tells you information about things on the linear genome, right? But of course, the DNA loops around such that distal regions can occupy the same physical space. So a few years ago, my colleagues Arup Chakraborty, Phil Sharp, and Rick Young in the biology department say that the way that they think about these so-called super enhancers is not just that you have multiple binding sites, but these multiple binding sites may help to nucleate a real physical cluster. Okay? And then they hypothesize that if these clusters form, they are likely to be very dynamic. So the individual molecules have to be able to quickly exchange. So they hypothesize that these clusters, if they exist, may have property of phase-separated condensates. And what most people understand by that is that it has a liquid droplet-like property. And I would say, when this was proposed, right, it's this idea that, was fairly controversial, right? Because there was virtually no information about how mediator is organized physically. So whether mediator clusters even exist inside a cell was not known. And the biophysical properties of this putative clusters, if they exist, and whether they would be consistent with phase separation, also not known. In fact, to our knowledge, even how Pol2 and mediator interact inside living cell had not yet been resolved. So that same year, in 2017, when this leading edge perspective came out in Cell, Rob had invited me to teach in a summer program that he teaches at Woods Hole. And during the summer program, you basically bring the wackiest idea that you can, because students are working extremely hard to try to solve some problem that they would not normally work on. And by way of doing that, they learn the techniques that you're doing. So I brought in my TAs, who is Wonky Cho and Jan-Hendrik Spiele, and I told them, look, why don't we try to address this problem and bring the problem to Woods Hole to try to address this. And so that's what we did. We moved to a type of cell called stem cells where people had done this ChIP-seq technique, so there is a lot of existing data. These cells like to grow close together. So in the same field of view now you can see multiple nuclei. And we labeled the endogenous mediator in these cells by a technique called CRISPR, and I don't need to go into that. Many of you have probably heard about it. It's a genetic modification that allows you to cut or paste anything. So if you cut the genome and then paste a GFP-like molecule next to the mediator gene, then that mediator will be labeled as well. So and then we can do live cell superresolution, and we can clearly see that mediator forms these clusters. And in fact, in addition to the clusters that we thought were big before, these ones form even bigger clusters. Again, remember that really, really big for me still means a couple hundred nanometers, right? So if we do TC-POM, we can see, in fact, that 90% of the mediator clusters in stem cells have this kind of transient clusters. And then about 10% have this kind of stable clusters. Now, these stable clusters, when we did it in Woodsall, the students in Woodsall decided to call them superclusters. And I asked them why in the end. So, you know, students there work extremely hard, right? They're working night and day. And they have 2 weeks to, you know, learn this technique and bring it home. And I think Muir from your lab, Muir Morrison, was one of those students, right? And so they told me later that they decided to call these things a supercluster because if they don't add the word super to it, our lab may not be interested in it, right? So they say we're doing super resolution imaging of super enhancers, so this stable cluster, they call it supercluster. And of course, you know, you have to understand, like, we had already seen that clusters can be stabilized by different ways, so I didn't think much about it. But the fact that they made me laugh about it made me think about actually using these clusters, and it was perfect. Because I would be honest with you. Earlier, when people will ask me whether the clusters we're seeing that last about 8 seconds have, you know, property of phase condensates, or that they're liquid-like property, I would say they last 8 seconds, so why does it matter if they're liquid-like? But now, if you have molecules that are very long-lived, then you don't need this highly specialized microscopy anymore. You can use more, you know, conventional microscopes. So it's still fancier than most, so we use a so-called lattice light sheet. But then you are able to visualize this molecule, and you can push for longer-term imaging than this light cell super resolution would have allowed. And what would happen long-term is that we can see these stable clusters diffusing around. So they're diffusing relatively slowly at the same rate as chromatin, so about 10 to the minus 4 microns square per second. But that means if we do tens of minutes long movies, we may find 2 of them diffusing coming very close to each other. So what should happen to 2 droplets when they touch each other? They should fuse into one, right? So we can test this directly just because this thing is slightly more stable and because we can push the microscopy with the lattice light sheet to much longer term. So that's what I'm going to show you next. It's a movie that is accelerated. You can see these things diffusing around, and when 2 of them touch, they in fact fuse into one. The movie will loop around, but then this is actually a 3D movie that we projected in 2D. So if you look in the X, Y, and XZ, you can see that they're in fact merging in all dimensions. So it's not just like one came and ducked under the other. And if you check the intensity, let's say for this orange and the blue, we're checking the intensities over time. In gray is the sum of the intensities. When they merge, they in fact go to the sum of the intensities, suggesting that they've in fact merged their content. There are many other properties that one can test. Remember that we're talking about droplets that form below the optical diffraction limit, so we are also limited on a lot of things that we cannot test. But there are some things that we can test. For example, you can ask whether individual molecules are going quickly in and out. So what do? You use a very high laser power to turn off all the molecules that are in the cluster, and then see how quickly they get replaced by molecules from the outside that you've not photobleached. And that tells you something about the exchange rate. So the molecules seem to exchange within just about 10 seconds. And then there are other things that block the interactions that are known to cause phase separation in biology. There is this drug hexandiol. People don't really know what that does, except it's known to melt a lot of liquid-like droplets, and it melts these ones as well. And then another thing that was known is that whenever I show you the picture of a protein, I show you a nice form picture. But for most of these proteins that are involved in transcription, they also have a flexible polymer that doesn't fold in a specific structure. And in phase transition in biology, it's this flexible polymer that actually are known to drive phase separation. So in many cases, you can just chop off these flexible polymers, purify them, and then prepare them in a test tube, and you can see them form these biological droplets. And we were able to collaborate with people in Rick Young's lab and show in this back-to-back paper that, in fact, this low complexity domain, as they're called, this flexible polymer, are sufficient to phase separate also in vitro. So moving forward, right, as a physicist, we know a lot about how droplets form, right? And, the example that I like to give students is that, me being from the Sahara Desert, right, in cold Boston, when I take shower, I take, really hot shower. So when I turn on the hot shower, then, what do you get? You get, the vapor, the supersaturated vapor that will condense, and for many key reasons, this condensation is better seen on a mirror, and you can go and write things like this on a mirror. I'm not the only one who does this, right? Okay. And we know a lot about how that happens. It happens through first-order phase transition, and a lot of the theory for condensation actually was thanks to this gentleman on the right here. So this one we easily recognize. What about this person? Szilard, that's right. So Leo Szilard here, reenacting the letter that, the sign of the letter that he and Einstein wrote to Roosevelt to start what would later become the Manhattan Project, right? Had thought a lot about how condensation processes work. And just to summarize a lot of careful work, the idea is that if you can figure out how to measure the free energy of these droplets that are forming and measure that free energy as a function of the cluster size, there will be the emergence of this nucleation barrier. So at this barrier, right, the first derivative of free energy is 0. That's the critical cluster size. Below this critical cluster size, things will fall down, so they will spontaneously disassemble by losing monomers. But above this critical size, things can actually stably grow by absorbing more and more monomers. Because of time, you can ask me to, later on to go through, some of, I think we have time, right? Like I have 5 minutes or so. Okay, so yeah, let's go through this for the students, right? So many of the students who have taken condensed matter physics will remember the work that is associated with forming clusters by classical nucleation theory in first-order phase transition. Is this kind of weird thing where it's a n to the 2/3 plus or minus bn. So for those who have not yet taken the condensed matter physics class, or who's been a few years, I would like to give some intuition of how you will build this kind of work. So imagine you're forming a cluster of size r, right? So the radius is r, and you're thinking about the energy that is needed to form this cluster. So you can divide the molecules in two species. The molecules maybe that are on the surface are just as likely to leave the surface as they are to stay. You'll need a positive energy term to keep them there, and that energy will scale with the surface, right? So it will give you something that scales with r squared. But then the molecules that are embedded, maybe they don't like entropically to be restrained there, in which case you'll need a positive energy. Or maybe, entropically, because of the system, you can talk about what that means later on, they are actually favored to form this condensed phase, in which case the energy will be negative and it will be minimizing. But either case, this energy will grow with the volume, right? So that will be something that scales with r cubed. In a condensate, you're forming a new phase at constant density. So the number of molecules that you get should scale with the volume. So n will scale with r cubed. You plug that in, right? If n is r cubed, then r is n to the 1/3. So here you get a n to the 2/3 plus or minus bn. So to dive a little bit more, this first term is nothing more actually than the surface tension times the area of the cluster that you have. And the second term is actually related to the change in the chemical potential for taking n molecules out of the solution and putting them in there. And so that depends on the concentration of molecules in the ambient case divided by some preferred emergent concentration for the system, the saturation concentration. So you get this NKBT, natural log of C over C saturation. If C is above C saturation, let's ask the student, then this ratio is greater than 1, right? And natural log of a number greater than 1 is positive. So then with this negative sign, you get a compensation between a positive and a negative term. If C is equal to C saturation, you get 1 natural log of 1 is 0, right? So then this whole term disappears. And then if C is less than C saturation, the sign flips. So that's why it's actually a phase transition at C saturation. So in a supersaturated state, that's C greater than C saturation, then you get a balance between this positive and this negative term. And that's how you get the emergence of this critical cluster size. So do we really mean that this could be happening inside a living cell? Well, for the transcription case, we know that there is a lot of biological regulation that may not, or that may hide the physics. So we can try to find out other cases where clusters can form, and then maybe that the cell doesn't care so much yet about regulating it. So if biology hasn't taken over, then maybe we can look at the formation process of this clustering and see whether this classical nucleation theory can explain how clusters can form in those cases. So the example that I'm going to show you is from clusters that form during Parkinson's disease. And we were able to capture with our microscopy that the very early stages of this cluster formation looks a lot like a condensation phenomena, the same type of merging mechanisms that I've shown with the transcription polymerase as well. So this movie will look around. So here you will get a three-way merger. This one merges with this here, and this one merges with this as well. And if you look at the intensity, you see purple and orange merge to go to their sum, and then when the third one merges, it goes again yet to the third sum. So then consistently what happens, and that I will not talk about, is that when you go to these very, very high cluster sizes, there seems to be an active mechanism that seems to regulate the cluster size and bring it back down as if there is some preferential cluster size. So maybe this will come to the question you were asking earlier. And this is actually something that Zillard predicted, but by just a theoretical mechanism, saying you cannot maintain supersaturation in steady state unless you have some kind of Maxwell demon that is looking at cluster size and then taking out clusters that get too uncontrollable. But he thought it was a theoretical exercise but in fact, textbook will tell you will never see that in nature, and I'm going to show it to you now. So what do we say? We say that the signal that we see, which should reflect how many detections we get, we check that it scales with the radius cubed of the cluster. And if that is true, and we are really able to look at the pre-nucleated clusters, the one even before they reach a critical cluster size, these are just molecules that are trying to climb over a nucleation barrier. There's a clear theoretical predictions for those ones. And it's just that it should be a Boltzmann distribution, right? You get the free energy, and you're trying to climb over that free energy. Once they cross over the free energy, then there is a lot of other things that can happen, including biological regulation, but also other more complex physical mechanisms like Ostwald ripening, right? The bigger will get even bigger at the expense of the smaller one. But we have a clear prediction about what should happen to the pre-nucleated cluster. So provided that we have the resolution, for this pre-nucleated cluster, this should happen. We just look at a snapshot of the cluster size, plot the distribution, the histogram of the snapshot, which should be the probability distribution. But rather than plotting the distribution, what we plot is minus log of the distribution and ask to what extent does that really look like a free energy. So you just take now minus log of a snapshot of these cluster sizes, you plot it in log-log form. And for the small cluster size, what should really dominate is this AN to the 2/3 term. And we can clearly see this kind of asymptotic limit towards the 2/3. So now you say, okay, let's accept that there may be this surface tension limit for the very small cluster size. What about we now subtract AN to the 2/3 from our data? What should be left? Now, many of us have done this, right, where we fit a data set and then plot a residual from that data set. And, we see something that is kind of squiggly. Of course, theoretically, what we should see is a line of negative slope. So I expected my student will come back and show me something that is squiggly and it looks like it's negative, and I would argue that, okay, it roughly fits like that. But this is what the data is. Strikingly linear. This told us that there might be many processes at play, but this simple classical nucleation theory in first order phase transition pretty well explains what is happening in the formation and growth of these clusters even inside living cells. So once you get all this, you know the two terms, and you know what the nucleation barrier is. The barrier seems to be about 7 kBt. So for us, that's really large enough to maintain a ratio of concentration that is 3 orders of magnitude difference, which is what we think is happening, right? Like, up to 1,000-fold higher concentration in condensed phase compared to the ambient phase. But it's small enough, let's say, compared to the energetic cost of 1 ATP hydrolysis. And if we look at what the critical cluster size, the size above which things are stable, it's about 160 nanometer, right? Intentionally almost below the optical diffraction limit, which could explain maybe why people may not have seen it before by studying this aggregate formation by conventional microscopy, but by super resolution you can. So this is where I really finish, and let me thank again. I've mentioned the group earlier, and all the super resolution data I showed you were really acquired and analyzed by Onkichio on the long-term imaging, especially the mergers, were done by Jan-Hendrik Spille, who built the lattice light sheet in our lab, and then the later work on classical nucleation theory, data acquired and analyzed by Arjun Narayanan, who really led the project. These were in collaboration with many different people. I mentioned the MBL Physiology 2017 class, the two-color super resolution, and especially the mRNA labeling with MS2 is really pioneered in the lab of, so the MS2 pioneered in the lab of Rob Singer, so we teamed up with people who trained under Rob Singer, Tim Leone, and Brian English, and Luke Labis developed the far red dyes for us that allowed us to do the two-color. And the later part on the mediator and enhancer work were in collaboration with people like Rick Young and Phil Sharp. I also acknowledge the physics of living system presence. The experimental labs are Jeff Gornick, the factory, and my lab, and our theoretical groups, including Lucia Kaborki, Leonid Bernie, Baron Kardar, and Max Tegmar, as well as these funding agencies who have really helped us take off on ideas that it was not clear would have a clear biological implication very early on. Thank you very much for your attention.
Time for a few questions. Michael?
Yeah.
So, the fact that classical nucleation theory works, I don't know, is there underlying assumptions there
Yeah.
that it's the interactions between subcritical sizes are isotropic?
Right.
It's van der Waals interactions?
Right.
or what? Yeah.
Right. So there are many implications. So what I didn't say, and that is actually quite important for this, at least the Parkinson-based, the tracer that we use is alpha-synuclein that later people find to form these fibers, right? But even when you go in the literature of the formation of these fibers, early on, people have hypothesized that the first steps before the formation of these fibers could be what they will call amorphous spheres, right? And EM early on would show that this was worked by the late Sue Lindquist at the Whitehead, and those were very purified alpha-synuclein. Here, we think what alpha-synuclein does is to go in clusters that form by many misfolded proteins that come from either defective translation processes or things that are already on the verge to being degraded. So we suspect that a lot of the clusters we're looking at are portions of proteins that are ubiquitinated, and that there can be a lot of transient interactions through that. Now, the fact that we can see this, n versus r cubed dependence really does suggest that there is some kind of isotropic interactions in building this cluster. Now, the final part, right, the final implication is that we are capturing this system at supersaturation, almost in steady state. And this was something that scared us until we started to read literature actually on Leo Szilard. So Szilard himself did not even write this down. Apparently, he would just sit in conferences and make this statement, and then other people go and write papers and credit him back on that. So Leo Szilard basically developed this idea that, okay, if you get this classical nucleation theory and the cluster reaches a critical size, then after that, then it's growing at the expense of smaller clusters, which means that the whole supersaturated state, I mean, I guess that's what the phase transition is, right? You're trying to deplete the supersaturation so that you now create this stable condensate and then a saturation level, C saturation, so that second term drops out. So, and then Szilard say, okay, that's horrible for theoretical calculation. So let's create this imaginary case that may never exist in nature, which is to say that there is a Maxwell demon that somehow is looking at the size of these clusters, and if it reaches some kind of aberrant size above that critical cluster, then you take it out of your system, and then you create new monomers to replenish. Then that will allow you a case where you're not depleting supersaturation, so you can have a steady state supersaturation, and you can start to write down something that looks like equilibrium thermodynamics and get predictions like a probability distribution. Well, a living cell is nothing more than Maxwell demons, right? And this was exactly the intuition that maybe if you go to neurodegenerative diseases, what goes wrong is that clearance mechanism. So things are already in supersaturation, so what goes wrong? And so we went and looked for one possible candidate, what would be this Szilard demon, and we think we identified it as well. Sorry, that was a long answer to your question. I believe there were a couple more questions, yeah?
But I was wondering whether you could relate the lifespan of the cluster to the dwell time of transcription factors that actually bind to those genes that you're looking at.
Right, so just to translate to the whole audience, the idea is that I'm looking at just the polymerase, but the polymerase is held by many transcription factors, so it's things like TF2D, TF2A, TF2B, TF2H that are believed to come in first, and we're working exactly on that right now to label these other things in different colors. So do dual color, not looking at the RNA output, but try to correlate that.
So a different explanation for why the cluster.
Exactly. So I already showed the mediator. In fact, we've shown even in this paper that there is a colocalization between the mediator and the POL2, and that depending on the transcription process, if you block the kinase that phosphorylate the low complex domain, you can purge out POL2 off and then wash it away and it comes back. So we are, in fact, looking at that. And it seems to be logical that there is other things in there that seem to control the residency. I believe there were two.
Maybe just one last question.
Okay.
You want to know?
You guys fight it out.
I do.
Go for it. Yeah.
It's a quick question. So with the super resolution, so if I understood correctly, the requirement is that things stay put, right?
Well, the requirements is that you're detecting things one molecule at a time. Yeah.
One molecule at a
Yeah.
time. Yeah, but something whether so in principle the things can move around.
Yeah.
And you can still associate.
Right.
With each other.
So that's right. So this is one key point about TC Palm. One thing that is really important, because in the end, we thought we may have to develop a completely new microscopy, but the same super resolution microscopy works. So what we can do is push the detection, like the camera acquisition time, to like a very, very, very high time frame. And for us, that really means what, like 20 milliseconds. I know other people will laugh at that. So the idea is that if the camera frame is 1 second and things move around, then you don't get a nice Gaussian, right? So you will not localize it very well. But if you push it to 20 or so milliseconds, then if things are transient even within that 20 milliseconds, so just a short period of time, then you get a much better localization. Now, of course, the number of photons that you get from that short time window is reduced, but maybe we're no longer trying to get the highest possible resolution in terms of localizing it, but just being able to tell whether it was there or not, right? And then the cluster size of 100 nanometer would be what we're trying to resolve. Yeah.
Yeah, I guess the comment is that
Yeah.
we can discuss more tomorrow.
Yeah.
Whether one can apply principles from control theory. When you have a model of how things move, you can incorporate it with the microscopy and then get
Absolutely.
traits like common control. Ideas.
Absolutely. So our main push, right, because this idea of doing super resolution live cell, I would say at the time, right, was quite controversial in a sense because people really believe that it takes such a long time to do super resolution that what are you trying to measure something that lasts just a few seconds? So we try our best to just tell people, we didn't do anything in the microscopy. It's exactly the same thing. It's just this time information that you throw away that we're using and then seeing things that you may have overlooked before. But now, that hopefully that's behind us. There is just so much room. Like as you say, there's control theory. There are ways of designing dyes, right? Like biochemists try to design dyes for single molecule microscopy that will last a really long time. Well, maybe now for TC palm, you don't want that. You want a dye that is super bright for a very short time and then goes away. So there are just so many different parameters that we can start to explore, both in terms of the data acquisition, the data analysis, and also the preparation of the sample to try to push TC palm. And I would be, of course, very happy to talk tomorrow about systems controls. Yeah.
All right. Let's thank Ibrahim again.
Thank you.